us humans seem to utilize it well, but you say it's also very important early on is sex. So, what is sex? Just asking for a friend. And when was it invented, and how hard was it to invent? Just as you were saying, and why was it invented? Why? How hard was it, and when?
I have a PhD student who's been working on this, and we've just published a couple of papers on sex. Yes.
What do you publish these? Does biology? Is it biology, genetics journals?
This is actually PNAS, which is the Proceedings of the National Academy.
So like broad, big picture stuff.
Everyone's interested in sex.
Yeah.
And the job of biologists is to make sex dull.
Yes. Yeah. That's a beautiful way to put it. Okay. So when was it invented?
It was invented with eukaryotes about 2 billion years ago. All eukaryotes share the same basic mechanism that you produce gametes. The gametes fuse together, so a gamete is the egg cell and the sperm. They're not necessarily even different in size or shape. So the simplest eukaryotes produce what are called motile gametes. They're all like sperm, and they all swim around. They find each other. They fuse together. They don't have kind of much going on there beyond that. And then these are haploid, which is to say we all have 2 copies of our genome, and the gametes have only a single copy of the genome. So when they fuse together, you now become diploid again, which is to say you now have 2 copies of your genome. And what you do is you line them all up, and then you double everything. So now we have 4 copies of the complete genome, and then we crisscross between all of these things. So we take a bit from here and stick it on there, and a bit from here and we stick it on here. That's recombination. And then we go through 2 rounds of cell division. So we divide in half. So now the 2 daughter cells have 2 copies, and we divide in half again. Now we have some gametes, each of which has got a single copy of the genome. And that's the basic ground plan for what's called meiosis and syngamy. That's basically sex. And it happens at the level of single-celled organisms, and it happens pretty much the same way in plants and pretty much the same way in animals and so on. And it's not found in any bacteria. They switch things around using the same machinery, and they take up a bit of DNA from the environment. They take out this bit and stick in that bit, and it's the same molecular machinery they're using to do it.
So what about the kind of, you said find each other, this kind of imperative to find each other? What is that? Like, is that?
Well, you've got to fuse cells together. So the bottom line on all of this is bacteria. I mean, it's kind of simple when you've figured it out. And figuring it out, this is not me. This is my PhD student Marco Colnaghi. And in effect, if you're doing lateral, you're an E. coli cell. You've got 4,000 genes. You want to scale up to a eukaryotic size. I want to have 20,000 genes. And I need to maintain my genome so it doesn't get shot to pieces by mutations. And I'm going to do it by lateral gene transfer. So I know I've got a mutation in a gene. I don't know which gene it is because I'm not sentient, but I know I can't grow. I know all my regulation systems are saying something wrong here, something wrong. Pick up some DNA, pick up a bit of DNA from the environment. If you've got a small genome, the chances of you picking up the right bit of DNA from the environment is much higher than if you've got a genome of 20,000 genes. To do that, you've effectively got to be picking up DNA all the time, all day long, and nothing else, and you're still going to get the wrong DNA. You've got to pick up large chunks, and in the end, you've got to line them up. You're forced into sex, to coin a phrase. So you're
so there is a kind of incentive.
If you want to have a large genome, you've got to prevent it mutating to nothing. That will happen with bacteria. So there's another reason why bacteria can't have a large genome. But as soon as you give them the power pack, as soon as you give eukaryotic cells the power pack that allows them to increase the size of their genome, then you face the pressure that you've got to maintain its quality. You've got to stop it just mutating away.
What about sexual selection? So the finding, I don't like this one. I don't like this one. This one seems all right. Like, what's is it, at which point does it become less random?
It's hard to know.
Because eukaryotes just kind of float around, just kind of have...
Yeah, is there sexual selection in single-celled eukaryotes? There probably is. It's just that I don't know very much about it. By the time we
you don't hang out with eukaryotes.
I do all the time, but
but you can't communicate with them yet.
Yeah, but for a peacock or something,
yes.
The kind of standard. This is not quite what I work on, but the standard answer is that it's female mate choice. She is looking for good genes. And if you can have a tail that's like this, and still survive, still be alive, not actually have been taken down by the nearest predator, then you must have got pretty good genes, because despite this handicap, you're able to survive. So
those are like human interpretable things, like with a peacock. But I wonder, I'm sure echoes of the same thing are there with more primitive organisms. Basically, your PR,
yes.
like how you advertise yourself that you're worthy
Yeah,
of.
absolutely.
So one big advertisement is the fact that you survived it all.
Let me give you one
good.
beautiful
Yes.
example of an algal bloom. And this can be a cyanobacteria. This can be in bacteria. So if suddenly you pump nitrate or phosphate or something into the ocean, and everything goes green, you end up with all this algae growing there. A viral infection or something like that can kill the entire bloom overnight. And it's not that the virus takes out everything overnight. It's that most of the cells in that bloom kill themselves before the virus can get onto them. And it's through a form of cell death called programmed cell death. And we do the same thing. This is how we have the different, the gaps between our fingers and so on. It's how we craft synapses in the brain. It's fundamental again to multicellular life. They have the same machinery in these algal blooms. How do they know who dies? The answer is they will often put out a toxin. And that toxin is a kind of a challenge to you. Either you can cope with the toxin or you can't. If you can cope with it, you form a spore and you will go on to become the next generation. You form a kind of a resistant spore. You sink down a little bit. You get out of the way. You're out of. You can't be attacked by a virus if you're a spore, or at least not so easily. Whereas if you can't deal with that toxin, you pull the plug and you trigger your death apparatus and you kill yourself.
Oh, so it's truly life and death selection.
Yeah. So it's a challenge, and this is a bit like sexual selection. It's not so they're all pretty much genetically identical, but they've had different life histories. So have you had a tough day? Did you happen to get infected by this virus, or did you run out of iron, or did you get a bit too much sun, whatever it may be? If this extra stress of the toxin just pushes you over the edge, then you have this binary choice. Either you're the next generation or you kill yourself now using this same machinery.
It's also actually exactly the way I approach dating, but that's probably why I'm single.